interesting chapter, and I don't know that we'll get all the way through it today. So our exam that was supposed to be on Wednesday, I'm probably going to bump back because I don't want to bust through the immune system and then in 2 days have to take a test over it. So whatever we don't finish today, we'll finish on Wednesday, and then I'm going to start respiratory. So we're going to play that whole game. Monday's a holiday, so that puts the test out till the next Wednesday. Which means you have lots and lots of study time.
The 27th?
The 20th.
Oh.
Next Wednesday, a week from this Wednesday. Monday the 18th is a holiday.
Okay. Got it.
Today's the 11th, I think.
Yep. Yes.
Are you a week ahead?
Probably.
Okay. So
Are
then,
we still having the IRAT on the
no. There will not be an IRAT on the 25th. There may or may not be another IRAT. Just saying. So then we got to look at what the rest going on with the rest of the semester. We have to cover respiratory. We have to cover urinary. And we really have to cover digestive. Reproduction's probably the one that's going to get tanked. So, I mean, like I said to somebody, when you're nursing, you spend a lot of time on all these major systems like you're following the kidneys and the urine output and ventilation and respiratory rate and all that stuff, and unless you're going into OB and mom and child care, whatever the hell they call that, mother and baby, whatever it is. Something had to fall. So if we get a few minutes, maybe we can look at a little bit of it, but I don't know, we'll see. So we gotta start chapter 15, which is our immune system. Those of you who are in micro, which includes a bunch of you, this will be review, which is good because the immune system can be a little tricky. So, I mean, we can spend a whole semester on an immune class. So we got a couple of terms to define here. An antibody is going to be abbreviated AB from here on out. AB for antibody. It is a protein. It's a protein, so it's transcribed and translated, which means it's the information comes from your DNA, and it is made by a specific cell called a B cell. We talked about white blood cells, and you guys identified them in lab. And a B cell is one of the lymphocytes. Remember, your lymphocytes have those big round nuclei. A B cell is one of the lymphocytes. The other type of lymphocyte is called a T cell, which we'll talk about in a little bit. So B cells make the antibodies. And the antibodies are produced in response to what's called an antigen. And an antigen is going to be abbreviated AG, and this is anything that initiates an immune response. A virus, a bacteria, a protozoan like Giardia, a worm infection. Even things that are not supposed to be antigenic, meaning they're not supposed to really look forward to the immune system, like pollen. Some people build an antibody response to things like that. But mostly we're talking about pathogens. Anything that is capable of causing disease. All those things that I just listed. So bacteria are single-celled organisms. Take micro, you'll learn everything you ever wanted to know or didn't want to know about bacteria. Single-celled organisms, they multiply on their own. Staph aureus, E. coli. Well, I can name a million of them, but the ones that I've named you probably never heard of. So, those are two biggies. Bacteria, those are the things that we can get infected with, we can get sick from. That's what antibiotics work on. Antibiotics work on bacteria, not viruses. So you need to take an antibiotic for a bacterial infection. not a cold. All colds are caused by viruses. Viruses are unique in that they are much, 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 much smaller than bacteria. They're teeny, teeny tiny. And they are made up of either RNA or DNA. And then they're covered by a protein coat. And that's pretty much it. So they've got the genes to make new virus in their makeup. And then they're covered by a protein coat. They cannot divide on their own. You cannot put a virus on some sort of a food source and it will divide. It can't. Bacteria, you can. You can put bacteria on a food source and it'll divide on its own. Viruses, no. Viruses have to be inside your cell or some sort of living cell for it to divide. Maybe you've heard of a tiny little virus called COVID. I mean, maybe, I don't know. The virus that, that's all we ever talk about now is COVID. It is just got RNA in it. It happens to be an RNA virus surrounded by a protein coat. Gets inside your cells, then your cell replicates the virus, and commonly what happens is the virus then blows your cell up and releases hundreds of other viruses to go infect hundreds of other cells, which then enter those cells, replicate to hundreds more, and it's an absolutely exponential growth. We can build antibodies to bacteria. We can build antibodies to viruses, even if we've never seen that virus before, just like COVID. Human population has never seen. COVID-19 was a novel virus. It was new, but you get vaccinated or you get sick, you build antibodies to it. Prions are unique in that they are infectious proteins, and we all have normal prions on our cell surface. They're normal, they're supposed to be there, they do a job. Infectious prions are ones that alter normal ones. So if our cell surface has prions that have a specific shape, because shape is vital to function in proteins, right? So these are all doing their job on the cell surface or inside the cell. And prions are abnormal proteins in which they have a different shape. And what's unique about them is that they sit down next to your normal one and they cause it to change shape, which then makes this one change shape and this one change shape. And then they don't work right.
Peer pressure.
That's right. And when they don't work right, then the cell dies. Right? Then I have normal functioning proteins. So, antibodies are a Y-shaped protein, and the foot portion of it, so they kind of like this, the foot portion is the constant portion. It's the hand portion that binds to the different antigens that changes. We have the capability of making antibodies to anything we come across. We have the ability to have an immune response and fight it off. This is the portion that changes depending on the antigen. So, if the antigen is this sort of shape, we'll make an antibody that binds to it. If it's a round shape, we'll make an antibody that specifically binds to that round shape. We've got that one, and that one, and that one, and that. It doesn't matter what it is. It doesn't matter the shape of it. It doesn't matter the contents of it. We have the capability to make antibodies to anything, which means we have the capability to fight any infection, as long as your immune system is working correctly. If your immune system is not working correctly, then you have trouble fighting infections. Antibodies are divided into different classes, and when we talk about them individually, then they're known as immunoglobulins. So the antibody is the umbrella term for all of them. When we talk individually about which class we're talking about, they're referred to as immunoglobulin, which just means an immunoprotein. Globulin is a type of protein. It's an immunoprotein. We don't say antibody G and antibody A. I don't know why. These are all the antibodies. When we're looking at them individually, they're IG. IgG, IgA, IgE for immunoglobulin. So we have 5 different classes of immunoglobulins. I'm skipping down, well, it doesn't matter. 5 different, this really isn't in your notes any specific place, this is just sort of extra. 5 different classes of immunoglobulins, 5 different types of antibodies. IgG are the most abundant. 80% of your antibodies are of the IgG type. They tend to float around in your bloodstream. They're secreted from the B cell in massive numbers, and they float around your bloodstream looking for that specific antigen to bind to. IgGs are very important in your immunity, your lifelong type immunity. It's these guys that are secreted in what's called the secondary response. The secondary response. In other words, you get, we'll say influenza or chickenpox. You get chickenpox as a kid, you fight it off, you build an immune response. If you're exposed to it 5 years later because you came across somebody's little brother who got chickenpox, you wouldn't get sick because you have immunity, right? The antibodies that are produced when you run into that thing again are of the IgG type. But most of the antibodies are of the IgG type. IgAs are found in secretions like tears, saliva, breast milk. This is the immunity that babies get from their moms from breastfeeding. So they say that breastfeeding is advantageous because the baby gets some immunity from it. Well, that's the immunity they get. They get some IgAs, which, you know, they're from mom, so they give the baby a little bit of an advantage, especially with the types of antigens that can enter the digestive tract and things like that. They don't last forever, so as long as when you quit breastfeeding, then these die off in about 2 weeks. IgEs, these are the ones that are in allergic reactions. These are the ones that people build that they build against things that aren't necessarily pathogenic. They're antigenic, meaning they activate the immune response, but we're not really supposed to be allergic to pollen, right? We're not supposed to have an immune response to pollen. That's not normal. That's abnormal. That's not something that is dangerous, pollen. But some people have allergic responses to them. Or to, it's usually susceptibility, genetics. Peanut allergies, nut allergies, seafood allergies. These are things that really are not pathogenic. They're not a problem for us. They're not going to kill us. But we sometimes build these antibodies to these normally undangerous, not dangerous, substances. What IgEs do is they stand on the surface of a cell called a mast cell or the basophil. Remember your basophil, that's the one with the big blue granules. Mast cells also have granules. And when the antibody comes across whatever you're allergic to, like pollen, then that binding to the pollen and the antibody activates the mast cell or the basophil, and then they release these granules that are full of histamine. And histamine is what gives you the allergic reaction. It's what makes your nose runny, and you sneeze, and itchy, watery. The stuff that I was going through this morning, that I had to go blow my stupid nose again. That kind of stuff. Whatever pollen's out there, I have IgEs that are recognizing it. IgMs, these are the guys that are important in when you first get sick with something. They're the first ones there. And they are made in massive amounts. Not as actually massive as the IgGs in the secondary response, but they're still made in big amounts. And what they're going to do is they're going to neutralize the antigen so that it can't, so that your immune system can take it over and get rid of it. IgMs are the ones that do not cross the placenta. All IgMs, let's do it this way. IgMs are the ones that are in your blood typing thing. So remember, if you're A blood, you've got A's on your red blood cells, right? And then you've got antibodies that recognize what? B's. They recognize the B blood type. These are IgMs. And IgMs are connected to each other in what looks kind of like a snowflake. It's a great big kind of mass of antibody. This is too big to cross the placenta. This is why mom can have A blood type and baby can have B blood type, and it's okay because the placenta blocks that size of antibody, and that's what this is. So in mom's bloodstream, she's got antibodies that would normally attack baby cells, right? But these antibodies are so huge, they don't cross the placenta to attack baby. Now, here's the question that we talked about in lab, and I said we'd talk about it later. If you are A positive, what does that mean?
Reese's.
You have the RH factor, right? You have the RH factor. This is where we can have issues with moms and babies. If mom is RH negative, mom's RH negative. Remember, RH antibodies are not naturally present in the bloodstream. We don't have those built up unless we see them later on in life. Let's say baby is B positive. We can make baby A positive. Doesn't matter. Should we make him A positive? Let's say he's A positive. That means that his little red blood cells have the A's and the RH factor. That's a new protein. That's antigenic to mom. She's never seen it. So when baby's growing, that's all well and good. Gives birth, these little red blood cells, because of the pulling away of the placenta and the bleeding, these little red blood cells get into mom's red bloodstream, and now she sees that protein, that RH, as something foreign, and she builds antibodies to them, and she attacks those red blood cells because they aren't supposed to be there. She's never seen that RH before. So now in her bloodstream, because she had a baby with that RH on it, now she's got RH antibodies. And these are of the IgG type. And they are single antibodies. They're not a great big snowflake.
So they can go through.
They cross the placenta. So now, it's not as much a concern for the first baby
As a second baby.
as the subsequent children. Right. As the subsequent children. I mean, it can be a problem for baby number one if there's been some, bleeding, some tearing of some mixing of the blood. But if there hasn't been until birth, mom's never seen that RH. So now baby number two, we'll say is B positive. Again, we don't need to worry about the antibodies to the blood type. We got to worry about that. Now she's got all these RH antibodies that are teeny, teeny, tiny, zippity-doo-dah, into baby.
It'll attack the baby.
And now these mom's antibodies bind to baby's little blood cells and target them for destruction. It's called fetal hemolytic disease. So mom is gonna get RhoDAM during pregnancy that blocks those antibodies from binding.
Is it in the butt?
I don't know.
Is that
I'm Rh positive, so I don't know.
Do the mothers get sick? Well, I just had a friend that her daughter had a complication between the blood types, and her daughter could not keep anything down. She was awfully sick, and it was because of the different blood types.
I don't know. That doesn't make sense to me. But
Okay.
I don't know. So if you are Rh negative, as a pregnant woman, you will always get RhoDAM. No matter what. Even if you can say, well, the dad is Rh negative as well. It don't matter. You're still gonna get
So what if you're positive and the baby's negative?
nothing.
Nothing happens?
Nothing. If you're positive, let's do that. Mom's Rh positive. So she doesn't have these. And baby's Rh negative.
Would the baby attack the mom?
Hold on. Baby's Rh negative. Little baby's red blood cells have Bs, but no Rh. So when these get into mom's blood, is she going to build any Rh or anything against it? No, because she's already got it, so we're not going to build an antibody against something I already have. When these blood cells mix with mom, will she destroy them with these? Sure, but who cares? It's not in baby, it's just a few blood cells that are floating around her bloodstream, right?
It's not enough to like harm her or damage because it's still there.
No. So this is not a problem. Positive, negative here is not an issue. It's the other way around.
If the mom's with the A positive, if it gets some blood in the baby that's being negative, can the baby then develop those proteins?
Would they develop the Rh antibody if they came exposed to moms? Probably yes. Probably yes. I don't know that.
Has occurred. So would that change the blood type?
Types of white blood cells, we talked about this in lab. Remember, our monocytes are the ones that circulate in the bloodstream. They've got the big horseshoe nucleus. When there is an infection and there's something that we need to go and get rid of, the monocytes squeeze out of the blood cell, or the blood capillary, squeeze out of the capillary and turn into a macrophage. And in the tissues, it's then phagocytic. Monocytes are not phagocytic, macrophages are. Lymphocytes, we talked about lymphocyte. Well, let's see. There's a picture of this. There's our phagocytosis.
So what would you describe phagocytic?
Macrophages.
Macrophages when they're squeezed out
Correct.
of the capillary?
Correct. Monocytes are not phagocytic, macrophages are. Monocytes floating around the bloodstream, just float around the bloodstream. It's only when they squeeze out of the capillary into the tissues to go fight something do they become phagocytic, and they get a name change. Lymphocytes have the big round nucleus. These are our T cells and our B cells. We'll talk more about those. And then we've got our granulocytes, the ones that have the big granules in them. Neutrophils have granules, although they're not very evident. They're not huge. But remember, the neutrophils have those pinched off nuclei, those weird pinchy nuclei. Eosinophils also have a pinchy nucleus, but what's more evident is their red granules. And then our basophils, remember, those are our big blue granules that release histamine. Histamine, let's see. We also mentioned this in lab as well.
Pam?
Yes.
Oh, we didn't go over IgG on the.
Didn't I?
No,
Okay.
sorry.
Let me go back to that. Let me finish our cells and then I'll go back. Thank you. Remember that monocytes make up about 8% of your white blood cells? About 8%.
Monocytes?
Monocytes. Lymphocytes about 25%. Neutrophils are the most abundant. Neutrophils are about 60-ish, 60 to 70%. Neutrophils are phagocytic. They go around and eat things. They love bacteria. Love bacteria. It's like their favorite meal. Neutrophils are always the first to arrive to a potential infection. Eosinophils are 2 to 4%, 1 to 3%-ish. 2%. They're involved in allergic type reactions. They can be phagocytic, but that's not their main job. They like to be involved in allergic reactions as well as like worm reactions. Like if you have a worm infection or a parasitic infection, you'll have lots of eosinophils. They can also be involved in inflammatory responses. They've got kind of a broad range. Basophils, those are the most rare. They're involved in those histamine allergic reactions, releasing tons and tons of histamine.
Like less than 1%?
Yep, less than 1%. Remember with phagocytosis, the cells that are phagocytic, mostly your macrophages and your neutrophils, they like to reach out and surround the antigen. In this case, it's bacteria. So they reach out, they surround it, they pull it in. Now it's in a little membrane-bound sac. They fuse it with the lysosomes. The lysosomes release their enzymes, chop it up, and literally exocytose it out the other side. Spit it out. And then we can reuse these components. It's like recycling. We can use those. They're the same things that we're made up of, right? Bacteria are made of the same stuff we are. Macrophages eat stuff too. Macrophages will eat viruses, bacteria. They also will eat dead cells. So if you've got an injury, your macrophages are the ones that are going to clean up the old dead debris. Neutrophils are more, their main source of food is bacteria.
So tattoos work as a adhesive and then holds it because it can't break down the tattooing. And so it just stays on the surface of your skin and then it dies and then when macrophages come up and eat it, and it just kind of hangs out.
Say that again.
So when you get like a tattoo, I believe it's the macrophages. They come up and they'll eat the tattooing, but they can't get it to break down. So it just kind of hangs out and they just hold it and then eventually they die and more macrophages come up and re-eat it, but again they can't break it down so it just kind of hangs out.
Okay. I will also suggest that that ink enters cells of the dermis, which is why tattoos don't slough off. So I don't think it's that that a tattoo only lives in the macrophages. I mean,
No, there was just a video that my physiology professor had, or teacher had me watch in high school. About it.
I've never heard that. All right, let's go back up here real quick because I forgot to talk about IgDs. IgDs are, their exact function is not known, not well understood. They believe that they stand on the surface of B cells and act as a receptor for whatever that antibody is specific for. So it's more of a receptor as opposed to a functioning type antibody. Now, the last cell here is a dendritic cell, and don't think this is a dendrite. This does not have an axon. Okay? This is not a neuron. Do not tell me that a dendritic cell is a neuron, because it's not. It's called a dendritic cell because these extensions look a whole lot like dendrites of a neuron. That's the only reason it has that name. A dendritic cell is also phagocytic, but it doesn't go out and eat lots and lots and lots of stuff. It's not a pig like neutrophils and macrophages. They just gorge themselves and then spit out their contents. What a dendritic cell does is it phagocytizes one thing, like a bacterium or a virus. Takes it to the lysosomes chop it up, and instead of exocytosing the contents, it takes the pieces of them and sticks them on the cell surface. Then what it does is it leaves the tissue spaces that it lives in and looks for one of the lymphocytes to tell the lymphocytes, hey, we've got this antigen running around here that could be potentially dangerous, we need to deal with it. So these guys are known as what's called an antigen presenting cell. They eat the one thing, I'm going to get rid of that because that doesn't live there anymore. They eat one thing, chew it up, and instead of spitting it out like a typical phagocyte, they take that piece and stick it out on the cell surface, and they go hunt for a T cell or a B cell, typically a T cell, one of the lymphocytes, to tell it there's something we need to worry about here. There's potential infection that's arising in the body, and we need to go fight it. Dendritic cells live in our tissues. They don't live in our bloodstream. They don't live in our lymph system. They live in places like the skin and the gut, the lungs, places like that. Places where we potentially see infectious agents.
Quick thing.
Yep.
How would you describe T cell and B cell?
Oh, hang on.
Okay.
So here's what happens in antigen processing, and I believe this is also presentation.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class I MHC molecules stimulate cell destruction by activating T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles, such as viruses and bacteria, by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
So I didn't get into that yet. When a dendritic cell chews up the thing, right, sticks it out on the cell surface, it sticks it out on the cell surface via a specific protein, either called MHC1 or MHC2. Don't freak out about which one goes with which, just MHC is fine. MHC stands for, it's on, I was going to talk about this in a little bit, stands for Major Histocompatibility Complex, which is right here. It's on the, when we talk about the T cells, it's on page 69 of your notes. Major Histocompatibility Complex. And the major histocompatibility complex is produced on all of your nucleated cells, all cells that have a nucleus, which includes every cell in the body, except for red blood cells, because they don't have a nucleus anymore. All nucleated cells have MHC, and they define you as you. So my MHC looks different than all of your MHC. And the cells that, your immune cells, learn that your MHC belongs to you during fetal development. So when you're a tiny little fetus and your immune cells are developing and your cells are developing, your cells, because of your genes, make MHC. And it's all different. Everybody's MHC is different unless you have an identical twin. Everybody's is different. It's all based on genetics. There's 4 different genes, multiple different types of the genes. Very, very complex. So your white blood cells run around the body saying, oh, that belongs to Pam. That's okay. I won't attack that cell because I know that that's supposed to be there. That's the body that I live in. If I were to get a kidney transplant from somebody else that isn't me, because I don't have an identical twin sister, then the MHC will be different. Now my white blood cells look at this and say, oh, that doesn't belong. Kill it. And they destroy that organ. It's called organ rejection, tissue rejection. We get rid of it, we kill it. So we don't want that to happen. If you're gonna get a new liver or a new kidney, then what you need to go on are immunosuppressive drugs so that this white blood cell doesn't notice that as easily. It dampens the white blood cell reaction so that you can't, it doesn't recognize it as foreign. And then we don't kill it and the person can live with a new kidney, but you are immunocompromised because your white blood cells are dampened down, which means you're more susceptible to other infections. You're more susceptible to getting sicker from COVID.
So do you have to stay on immunosuppressants?
Yes.
Forever? So what is the testing that they have to do to see if an organ is like compatible?
They used to do tissue typing, and as I understand it, you don't really do that as much anymore because these drugs are so good at dampening the response. So as long as you have the right blood type, so all they do is look at blood typing, pretty much. They don't look at the tissue typing anymore.
Wouldn't tissues be better almost that we were not on drugs long term?
Oh, sure. Sure. You would be better if you didn't have to be on an immunosuppressive drug, right?
Then why did they stop that? Money?
Because to get it perfect, unless you have an identical twin, it's never gonna be perfect. And this is easy. So here's the other issue. If you need a new liver and you're waiting for the perfect match, odds are you're not gonna get it because you're waiting for the perfect match. Now, we can give you a liver from pretty much anybody in the room that's got your blood type, and as long as you take the drugs, you have a new liver and you'll live.
So
Makes
what
it
happens
easier.
if it's not from the same blood type?
You can't do it.
It just attacks it.
You can't do it. And I don't know why the blood typing thing is such a big deal. But that's one of the requirements, is the same blood type. But not necessarily the same MHC. You can have a totally whacked out MHC and it'd still be okay.
What about people who are like O negative who can donate blood to anyone? Does that go for organs as well, or does it actually have to match?
I don't know. I'm gonna guess. I don't know. I was gonna guess, but I'm gonna change that answer to I don't know. I don't know. I have to look into that, but I'm not sure. All right, our immune system is divided into two sides. There's two sides to the immune system. It's the same chart that I show in micro because I like it. We've got two sides of the immune system. We've got innate immunity, and we've got acquired or adaptive. I don't like either of these terms. Innate immunity is the non-specific immunity. Acquired immunity is specific. Okay, so we've got two sides here. Innate immunity or non-specific immunity has two parts to it. We have the first line of defense, second line of defense, and if we scoot over here, this is our third line. Okay? First and second lines of defense are part of the non-specific side. This means that our immune system is going to go out and going to non-specifically fight whatever shows up. If you get a skin infection or an abscess in your tooth, it could be full of tons of different bacteria, right? This side of the immune system will go out and attack all the different types of bacteria. They're not just going to go do one. They're going to get everything. They go and they fight non-specifically, whatever they're going to go get. The specific side, the acquired side, is the side that's going to fight one infection, like COVID, or influenza, or chickenpox, or Staph aureus sepsis, whatever. It's one thing, one specific infection. So, first line of defense is the physical barriers. Oops. Physical barriers that keep the pathogens out. Skin is a perfect first line of defense. It's the best protection against infections out there. It's thick, it's got a bunch of dead cells that slough off, it's dry, it's a little bit acidic. As long as it's intact, nothing's going to get through. Bacteria don't crawl through your skin, viruses don't crawl through your skin. As long as it's intact, nothing gets through. If it gets broken, then we have the potential for infection, right? You get a cut, potential for infection. You get a scrape, you get a wound, potential for infection. Another physical barrier is your mucous membranes, any tube that opens to the outside, which makes tons and tons of mucus. That mucus lines the tube. It's a thick layer of mucus that blocks bacteria and viruses from getting to the cells, whereupon if it's a bacterium, they have to stick to your cells to divide. If it's a virus, they have to enter your cells. So if you have a big, thick layer of mucus, maybe they can't get into or swim through or move through the mucus to get to your cells to infect them. Many of those mucous membranes are also ciliated. Not all of them, but some of them are, like your respiratory, especially your respiratory tract. So your respiratory tract, which is open to tons of infections, right? There's viruses from people sneeze, that's how you get COVID, you breathe it in, and then it gets into your cells. So your ciliated, your respiratory tract has cilia, which beat unidirectionally, either upward to your throat to be swallowed down or coughed out, or backward to your throat to be swallowed down or coughed out. So that helps move that mucus, which keeps the pathogens moving, and hopefully you can get them out before they stick to your cells. Acid, the stomach. If you do eat something that's pathogenic, if you think everything you eat is sterile, you're out of your tree. You are lost in space. Nothing you eat is sterile. So your stomach has a pH of between 1 and 2. We've talked about that. What does that do? What's that high acid do? What does it do?
Kills anything
How?
by breaking down
How?
through the acid.
How?
Disintegration.
What?
Like disintegrating it?
Nope. Anytime your pH is out of physiological norm, what happens?
It can burn a hole through your stomach.
Oh, breaks down the protein.
It does what?
It ruins the DNA. It
Denatures proteins. Denatures proteins. When proteins get denatured, the cell doesn't work right, right? It doesn't work. It blows open. It dies. So, you eat potentially pathogenic things, they go into your stomach, gets denatured, nothing. So, physical barriers, what else do we have? Other places of acid, the vaginal canal, it's acidic, denatures proteins. Kills potential pathogenic organisms by denaturing proteins. Tears, saliva, we're always washing our eyes with our tears. It drains from here into the corner of the eye, which ultimately ends up in your nose, which then you can either sniff up and swallow down or blow out in a Kleenex. Saliva, we're always making saliva. Saliva washes bacteria from your mouth. Not all of it, but it helps. And that goes down into your stomach again for more denatured.
Is this all first line of defense?
Yes. Correct. All first line of defense. Now, if your first line of defense is broken, then we've got something we have to do here. We've got to work through our what's called inflammatory response. Anytime you've got damaged cells, you're going to have inflammation. Because what happens is, the cells that are damaged release cytokines. We talked about cytokines before, right? Chemicals that are released that cause an action in another cell. And these cytokines, things like histamine, makes blood vessels leaky. If the blood vessels are leaky, then those macrophages and neutrophils can squeeze out of the blood vessels more easily and fight the infection. The cells that are damaged that release these chemicals also release cytokines that are inflammatory. Inflammatory can cause damage to other cells. Inflammation's not a good thing. We don't want a bunch of inflammation in our body. But this happens if we've got an injury. You have swelling, you have heat, you have redness, you have trapped white blood cells, etc., etc. This is a normal response. Some people have abnormal inflammation, and that's a bad thing because now these chemicals are being released all the time, and that can be dangerous to the body. So inflammatory response is a good thing if it's held, in a local, but if it's widespread, it's not good. So here's a picture of the inflammatory response. This person was poked in the skin with a very, very, very dirty pin. We've got damaged cells. The damaged cells release cytokines. Those cytokines attract the phagocytic cells that are in the capillary, It's like these phagocytic cells are just scooting along here in the capillary and they smell the chemicals. When they're attracted to those chemicals, it's called chemotaxis. Chemotaxis means attracted to a chemical. So they smell this, kinda. These chemicals also make the capillary more leaky. As they're more leaky, it makes it easier for the phagocytic cells, your neutrophils and your monocytes, to squeeze out. Monocytes turn into macrophages. Neutrophils stay neutrophils. The neutrophils go and eat the bacteria. Macrophages eat bacteria too, but they also come along and eat up the dead cells that are, no longer functioning. We also have injury to blood, possibly injury to blood vessels. So we've got to secrete our clotting proteins. We've got to do the whole clotting thing. So we'll stop the blood flow. Your macrophages are out there and your phagocytes are out there chewing everything up, cleaning everything up. We've clotted the damage and hopefully taken care of this infection before it could spread to farther locations. This is non-specific. It's part of the non-specific response. It doesn't matter if this is Staph aureus, E. coli, Proteus vulgaris, M. luteus, Staph epi, B. sub, A. faecalis. None of those would be there unless you got poked with a pin that was stuck in poop, right? Just saying. They're the only ones I can think of off the top of my head. But all of these are different. Let's say they're all different. Does this neutrophil or macrophage care? Does this one say, well, I'm only going to get the staph. That's all I'm going to go after. We're not going to worry about the rest of those.
Third line of defense.
That's right. Now we don't care. Non-specific. They're going to eat them all. We want to keep this, if this is going to be an infection, we want to keep it local. We don't want it to go widespread. Can it go widespread? Sure. Then it's called sepsis or cellulitis or, more spreading of the infection. So what the job here is to take care of it quickly, non-specifically, just go out and get rid of whatever it is. First line of defense, these are the guys that are out there working hard, keeping everything small and tiny. The cells that are in charge here, the cells that are in charge of the second line of defense are your phagocytes. Phagocytes rule the second line of defense. We don't have any cells in the first line of defense because that's your physical barriers keeping it out. If your first line of defense gets broken, then we've got our inflammatory response and the phagocytes come in and eat it all up. These include our neutrophils and our macrophages. Another part of the second line of defense includes something that is going to attack bacteria only, and it's called the complement system. So inflammatory response is one part of the second line of defense, another part is complement, and this is bacteria. Fights bacteria only. So complement is a series of 9 proteins that circulate in the blood in an inactive form. They're just floating around. And if you've got a bacterium, typically, that is surrounded by some antibodies, then complement comes in, cross-links 2 of the antibodies, and then calls on all of its other 8 neighbors, friends, 1, 2, 3, 4, 5, 6, 7, 8 of them. And what they do is they insert themselves into the surface of the bacteria, forming holes, and all the insides fall out, and the cell lyses. So this is a bacteria thing only. Complement can be activated directly by binding to the organism itself, but it's easier if some antibodies have already bound to the bacterium. It just goes a little faster. First line of defense keeps it out, blocking the pathogens from getting in. If they get in, we have the inflammatory response. Who rules the inflammatory response? Phagocytes. We also have complement to attack bacteria only, poking holes in them, blowing them up. Now we're going to look at the third line of defense. Third line of defense has two sides. We've got the antibody, which we do first. That's fine. Got the antibody mediated and the cell mediated side. Antibody mediated means we're gonna make a bunch of antibodies. Who makes the antibodies? B cells. So the B cells run the antibody mediated side, which means who do you think is going to run the cell mediated side? B cells. Correct. So we'll start with the B cells and we probably won't get to the T cells today. B cells. B cells make antibodies. We already talked about the different classes. Antibodies are not like little grenades that bind to, well, let's say this is a virus, funky looking virus, but whatever. Antibodies recognize foreign things. They bind to antigens. But they are not a grenade that blows it up. They do not kill. Antibodies don't kill. All they do is they bind to the antigen and surround it. And what that does, I gotta get rid of that because that's not what happened. What that does is targets it for destruction. So when an antigen, bacteria, virus, is surrounded by antibodies, that means that that antigen is opsonized. Opsonized. Opsonization means it's surrounded by antibodies. Surrounded by antibodies. If it's surrounded by antibodies, then your phagocytes see it, recognize it, and eat it faster. They'll eat a virus floating around. They'll see it as foreign floating around and they'll eat it. But if it's surrounded with antibodies, it makes it much, much better. Makes it yummier. What should I say? Let's see. They usually use brownies. Let's use cupcakes. Right? Cupcakes are good. Unfrosted cupcakes. Would you eat an unfrosted cupcake? Sure. Chocolate, chips, something like that. Macrophages eat them too, so do neutrophils. But if you put frosting on, like antibody frosting, yum, 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 makes it much more delicious. So they eat it faster. So think about opsonization. Think about antibodies as frosting on the antigen, which means your macrophages thinks it's better. Your neutrophils like to eat it faster. What else do antibodies do? If they surround, let's say this is a virus. If the virus is surrounded, if it's covered by frosting, it's covered by antibodies, I said that in order for a virus to get replicated, what does it have to do?
Go into
It has to get inside the cell. Well, if it's covered by antibody frosting,
you can't go in.
it can't get in, and it ain't gonna get replicated. It's gonna get eaten by a macrophage. Those are the two main things. Those are the two main things that antibodies do. They surround to block or to make it better for your phagocytes to eat it. They surround to block binding and infection. B cells. B cells make antibodies. B cells make antibodies. But in order to make the antibody, the B cell needs to recognize an antigen and then divide and become a cell called a plasma cell. B cells are floating around. They're floating around. They're actually hanging out mostly they're hanging out in your lymph system, sitting there. And if they recognize an antigen in one of a couple of ways, either the B cell can have little receptors on them that the antigen binds to, or a antigen presenting cell who's got a little antigen sticking here out on the cell surface can show the B cell, hey, look it, I'm presenting you, I'm showing you this piece of antigen, and I'm telling you there's something we need to make antibodies for. So the B cell has to recognize the antigen in one of a couple of ways. Either it does so directly, or it gets presented the antigen. Once it gets presented the antigen, it needs to divide and turn into a bunch of cells called plasma cells. And plasma cells spew out antibodies. Tons and tons and tons and tons of antibodies to that one thing. 200 or 2,000 per second. So you divide into tons of plasma cells who make 2,000 antibodies per second. So how many antibodies do you think we're going to make in, a day?
Loads.
Tons, right? And then what are these antibodies going to do?
Surround it.
Frost. They're going to frost stuff. They're just going to cover things. That's all they're going to do. They're not little grenades. They're not little missiles. They don't go blowing things up. They're going to frost. Okay? Here's the kicker. A B cell recognizes the antigen one of two ways. Either binds to it directly or it gets presented. But the B cell can't turn into the plasma cell until somebody tells him he can do it. So he recognizes the antigen and then gets stuck. It's like, oh man, there's a battle out there. I gotta make a bunch of antibodies. I gotta do it. You can't do it. It's like he's tied up and he can't go. Somebody's got to tell him that he can go ahead and divide and become plasma cells and make a ton of antibodies. And the guy that's going to do it comes from here. One of the T cells is going to tell the B cell, all right, I know you want to turn into plasma cells. I know you want to make a bunch of antibodies. But until I tell you you can, you can't. So, T cell side, we have to wait. We have to wait for the T cell side to tell the B cell side what to do. We you have to wait for the T cell side to tell the B cell side what to do. Did I say that right? Or did I say it backwards? So who are the T cells? Well, we've got several. We got several types of T cells. One of the T cells, let's go to the next page. One of the T cells that I talked about first is called a natural killer cell. NK cell. NK cell is a lymphocyte derived, it's a T cell derived cell. He really does his work over here. He's really out there working all the time. But we talk about him over here because that's kind of where on the lymphocyte side. NK cells are the patrollers. They're the scouts. They're out looking for trouble. They're floating around the body looking for trouble. What do they look for? They recognize cells that are infected with a virus and cells that are becoming cancerous. When cells become cancerous, their genetics get messed up. Their MHC proteins that normally we recognize as okay get messed up. Cancer cells' genetics are all whacked up. Once they start dividing, they divide too quickly, the chromosomes don't separate right, they're just a mess. So these NK cells, one of their jobs is to run around and look for potential cells that aren't supposed to be there, that don't look like you. They also look for cells that are infected with the virus. So if you have a cell and the virus got inside, this cell says, oh crap, I'm gonna die soon and there's gonna be a whole bunch of other viruses that are gonna be released and kill a whole bunch of my friends. So what I'm gonna do is I'm gonna put a little white flag out on the surface of my cell that says, I'm infected, it's got problems coming. And so what the NK cell does is he comes along and he sees that and he shoots it. Shooting the cell, killing the cell. The only analogy I can ever come up with, and I can never change it, so your micro people are gonna have to listen again. The NK cell shoots the infected cell with a shotgun type shot. And how do shotguns go? So this guy's not infected, and that guy's not infected, but because the bullets spray, guess what? We got some collateral damage. Right. They're gone. Which is okay. I mean, it's sad. It's a sad story. But this guy who's infected with a virus, remember, when viruses get replicated in your cell, you can make hundreds of new viruses. Tens to hundreds. Say this cell, if this cell didn't get killed by the shotgun, 100 viruses would have been released. How many cells are going to be infected? 100 more. And of 100, if they all release 100 viruses, then how many do we have? 100 times 100 is then 1,000. And then those 1,000 cells get infected and release, each one releases 100 more, right? So if we can dump that dude before he releases these, I'm sorry, you guys didn't make it either, but you know, it's life of the war, right? So we got a little collateral damage, but it's way better than the thousands that are going to be killed in the next day.
So what happens when the virus spreads?
The other cells get blown up.
Yeah, but is the NK just not finding it fast enough, or?
They're working hard. They're working hard. Then we gotta get the rest of the big dogs in place. So think about like when you get a sore throat, like you get a cold and you're getting a sore throat. First it's scratchy. Those are a few cells being blown up. Maybe a few NK cells out there. And then some more viruses are released, more cells are infected, more cells are blowing up, the NKs are out there just shooting like crazy, and then the next day your throat is really, really sore, because it's raw. The lining has been blown up by viruses and NK cells. Then, you gotta get the rest of this story, which will be on Wednesday. So we'll finish this up on Wednesday. I will start respiratory. But remember that won't be on the test.